Imagine being able to edit, copy, and modify the building blocks of human life itself to cure diseases, enhance our bodies, and potentially change the evolution of human life itself. CRISPR is a gene editing tool that rewrites the rules for biology as we know it and allows us to do that. Understanding how the CRISPR process works, the applications, the potential applications that it may bring, and also the ethical concerns that could potentially come with it is important for understanding why CRISPR is the future of science as we know it. The first step, understanding how CRISPR works, is important. What happens is the first step in CRISPR is identifying the target DNA. Then a guide RNA is created by researchers. The guide RNA serves as specific RNA that matches with the target sequence and directs the Cas9 to the precise location for editing. And then the DNA break occurs, and the enzymes in the cell work as natural repair systems, stitching the DNA back together. Some potential applications. Some potential applications for CRISPR are disease treatment by targeting defective genes and editing them. An example of this is the use in diabetes care with beta cells transplantation into diabetic patients, where the genes in beta cells are modified so that the immune system does not immediately recognize the insulin producing beta cells, because diabetes is basically when the immune system kills the insulin producing beta cells, making it harder for diabetics such as myself to produce it. Another use of CRISPR is conservation genetics that are used to preserve endangered species. Editing the genes of animals that may not be able to survive in the current environments with new diseases and new environmental changes is really important, and it would allow them to survive longer. And CRISPR can also be used in biopharmaceutical production to create new vaccines and antibodies more effectively and increase the consistency in cell lines. Some ethical concerns are definitely that it's a very new technology, and this can produce a lot of off-target effects or unintended genetic changes that can potentially lead to consequences because we don't know much about it yet and haven't done it on many test subjects. Editing the genes of the germline also gives a lot of ethical concerns to future generations and current generations alike, as people are really questioning, the humanity of children that are able to be created from, basically edited from birth, and people can decide what kind of babies they have, decide what kind of babies they have, almost a make a baby, if you want to say it per se, and that's not good. And a lot of people are questioning if you should be able to choose the qualities of your child. And these technologies are also going to be very expensive and difficult to access. The healthcare field as it is right now is already terrible, and people are usually not able to access what they need to access at a reasonable price and within their price range. And these future technologies may cost hundreds of thousands of dollars that most people can afford, even if they need it, and it may make their lives better. So that's going to be another debate. In conclusion, CRISPR serves as a revolutionary achievement in science that has the power to revolutionize multiple fields and, most importantly, change how humankind lives. And understanding how CRISPR works is very important, as it's the foundation for grasping its effectiveness. And also, the precision will allow people to address more diseases, address genetic disorders, have better agricultural practices, and ultimately understand life's process as a whole.